Hello, everyone. My name is Aayushi Uberoi, and I'm an assistant professor in the Department of Pathology and Immunology at the Washington University School of Medicine at St. Louis. Today, I'm going to talk about my body of work in which we basically discovered that commensal microbes can be used to fortify the skin barrier. When we think of the skin, it is one of the first lines of defense which protects the host against microbes, pathogens, and environmental exposures. So, histologically, if we were to take a human skin section and see it under a microscope, we would see something like this, where you can see these distinct layers that the skin has. In purple is what we call the epidermal layer of the skin, where you see this wonderful pattern in how the cells differentiate to give rise to this outermost layer, which is called the stratum corneum. And then at the bottom, in this below this line of demarcation, which is in white, you actually see the dermis, which is consisting of the fibroblast layer. Now, what we have understood from all these years of dermatological research is that why the skin is such a fascinating stratified epithelia is that each layer of the skin has been programmed into behaving a certain way. So, for example, if we were to look at just the basal layers of the skin, as shown here in this immunohistochemistry, we can actually see that they specifically stain for cytokeratin 14 markers. And then as we stain them with cytokeratin 10, we see that everything except that layer stains. The point of this is that epithelial differentiation is a tightly controlled process, which you can see being depicted here, where we want that under homeostasis, all our skin cells know how to behave properly in the sense that this pattern is followed. And this process is called epidermal differentiation process. For most part, in a state of intact barrier state, or what we call a homeostasis state, we should see all these layers being extremely distinct. And then we should also see a reticulate pattern, which you see right on top, this web-like interface in dark purple, which is called the stratum corneum, and that's the topmost layers of the skin. At this stage, we would call that the skin has an intact barrier, meaning it is protecting the host from different external influences. Now, if there is a compromised barrier state in which you can actually see here is a very adverse circumstance in which the topmost layer has been completely removed. Now, at this point, the dermis and subsequently all the underlying organs below the skin are actually susceptible to external insults. And this is a state of compromised barrier. Now, compromised barrier is implicated in a variety of diseases. So, for example, in homeostasis, where we see this intact disease, if we were to completely lose the barrier, we would get a barrier dysfunction, which is implicated in atopic dermatitis, psoriasis, and other diseases. We could also have an acute barrier defect in which we basically would see what is in parallel with wound healing responses, where if there's an acute wound, we would see a compromise of the barrier again. And in some cases, this differentiation process can get completely altered, in which case we would see papillomatosis. Now, for most part, this whole aspect in which we can control how the skin moves from homeostasis to these different disease states is regulated through the host. But there is an important component which also regulates these processes, and that is the skin microbiome. So, skin, along with all of this complex cellular architecture, is also home to the skin microbiome or the SIT, and is therefore called the skin ecosystem. And why this is relevant is that the skin microbiome is extremely complex and diverse. So, for based on just the biogeography of the skin, we see that oily areas such as the forehead, etc., have like low diversity, which is depicted through a lot of species like Cutibacterium and Corynebacterium. Whereas if we move to the more dry parts of the skin, which would be, for example, the forearm, you would see Proteobacteria and Bacteroides. But the point being that the skin microbiome is both diverse and topologically diverse as well. It is extremely distinct from other barrier organs. So, skin microbiome communities are completely different when compared to gut microbial communities or vaginal communities and other barrier organs such as the respiratory tract as well. But what is critical is also that although the last decade of skin microbiome research has really focused on elucidating the composition of the skin microbiome, it is extremely understudied microbiome in terms of its functional contributions. But what we do recognize now is that skin microbiome is a functional unit of the skin barrier function. So, in its most primitive form, the skin microbiome basically fills these ecological niches which prevent pathogen colonization. So, by the virtue of basically fighting against microbes and occupying niches, skin microbiome, which is the commensals, can actually fight against pathogens from invasion. What skin microbe commensals can also do is they can uh, basically also educate and prime the immune system barrier. It has been shown through decades of work in immunology that the skin microbiome communities are seminal in educating and priming the immune barrier and basically also priming the neonatal immunity aspects of the skin. However, what is completely not understood is there any impact of the commensal skin microbiome on the physical barrier of the skin? And by physical barrier, what I mean is this ability of the skin microbiome to regulate the skin's ability to regulate barrier permeability. So I explained how skin serves as a barrier against external insults, but skin by itself also controls the hydration parameters of the rest of the body. So if we were to not have an intact skin barrier, we would basically get completely dehydrated. And this water balance is extremely critically maintained by the skin. And this physical barrier permeability aspect resides in a few properties of the skin. So, by an outsider perspective, the physical barrier is regulated through the keratinocyte differentiation program. And basically, this function resides in the stratum corneum of the skin. And then it is also regulated by how tightly these keratinocytes are packed, which is marked by cellular adhesion junctions. What I'm going to tell you today is how we found that basically skin microbiome regulates this epithelial barrier permeability function of the skin. And then I'm going to delve into what are some of those mechanisms by which skin does that. So we found that skin can do so through regulation of the aryl hydrocarbon receptor, and it can also do so by certain properties of the commensal microbes. And exactly what some of those mechanisms are is also something I'm going to talk about. So, this work started out with the fundamental question that exactly what are the microbially regulated pathways in murine skin? And this was done because I wanted to understand exactly what are the functional contributions of the commensal microbes. And I made use of the powerful tool of germ-free mice, which are raised and generated in the absence of microbes, and basically compared the transcriptomically regulated pathways with that of specific pathogen-free mice, which are raised in presence of microbes. So when we specifically did epithelial transcriptomics, which basically involved taking keratinocytes from these mice, we found something very interesting that commensal microbes regulated expression of barrier genes. This was extremely surprising. So the genes that we saw were mostly regulated by commensal microbes. Some of the top genes were involved in skin development, keratinocyte differentiation, and epidermal development, which suggested that basically commensal microbes are seminal in regulating a very fundamental aspect of epithelial differentiation. When we look at the histopathology, such as of these mice under this microscope, We don't really see a remarkable difference stand out when we look at specific pathogen free mouse skin versus that of germ free skin. However, if we do electron microscopy on these sections, we can actually see that SPF mice have a greater number of stratum corneum layers. If you look at this figure closely, you will see that the germ-free mice in some parts are even lacking some of these layers. And if they do exist, they're extremely loosely packed. As opposed to the SPF mice, where we see this pattern to be completely beautifully connected and almost looking like a wall of bricks, which is well protecting us from the external environment. Now, I told you also that a lot of genes that we saw were implicated in keratinocyte differentiation. So, if we were to stain protein markers specifically, what we found is that germ-free mice showed a downregulation of some of these genes even at continuous light exposure compared to SPF mice. If we look at how the keratinocytes are packed, we can stain for a gene called desmoglein 1, which basically stains tight junctions and adherence junctions of these skin cells. And we see that in SPF mice, as you can see, there's a beautiful reticulate pattern of desmoglein staining, whereas in germ-free mice, that is extremely downregulated and almost diffuse. Another thing that we did was we looked at the epithelial resistance properties of keratinocytes derived from these mice. So, a test which is used in the field is basically to measure epithelial electrical resistance, in which we can test barrier function. In the skin, this assay is not so easily adaptable because it is a stratifying epithelia. However, if we take keratinocytes and stratify them in presence of calcium, we can measure this epithelial resistance because now we can polarize the cells. So here, what we would expect is that if there's an improved barrier state, we should see increase in epithelial resistance. Whereas if the barrier is weakened or compromised, we should see a decrease in epithelial resistance. So if we take cells which are derived from germ-free mice and specific pathogen-free mice, to my surprise, what I found was that the germ-free primary mouse keratinocytes were impaired in barrier function. So if you can see in this figure in blue are cells which came from germ-free mice, and over several days of calcium-induced differentiation, we actually never see the epithelial resistance reach where it was for SPF mice. If we specifically take these primary keratinocytes and look for proteins through quantitative gene expression of genes which are implicated in epithelial differentiation, we continuously see that those genes are downregulated. So, for example, genes like keratin 10, involucrin, and even those implicated in adherence junctions are impaired in germ free mice when under high calcium expression conditions. So, at a molecular level, hopefully, I have convinced you that commensal microbes do regulate a lot of fundamental properties of differentiation of keratinocytes. But what does that mean functionally in terms of the health of individuals or animals? Is what we really wanted to understand. That what are the implications of these findings? So we borrowed, basically collaborated with veterinary dermatologists where we can adapt an assay which is used in dermatology clinics, which is that we can measure transepithelial water loss at the skin surface. This basically helps us assess how well the skin is controlling the water loss function, which is a direct assessment of the barrier permeability. This assay is often called tool, as I will refer to it in this talk. But the concept here being pretty simple, that if there's a healthy skin environment, the skin should be able to control how much water we see at the surface. Whereas if it's a compromised barrier, we will see increased water loss, which will be marked by an increase in tool. I adapted this assay to basically compare barrier recovery kinetics in germ-free versus SPF mice. And what that means is that to assess barrier repair kinetics, I basically conducted tape stripping on the skin of germ-free versus SPF mice, where they were compromised, the barrier was compromised using tape strips to the same consistent level. And then over time, transepithelial water loss was measured. How this would look really in terms of a schematic would be that at time of tape stripping, we should see a increased barrier loss and increased water loss at the surface. So we would see an increase in tool. But with time, as the barrier repairs, we should see a decrease in water loss, indicating repair of the skin barrier. So for most part, this works really well in SPF mice, where when we perform a tape strip, which you can see at time zero, we see an increased water loss, whereas over 24 hours, as we continue to measure the tool function, we actually see that the barrier repairs itself and we see the tool levels improve. When we look at germ-free mice, we find that they are slower to recover. So hopefully you can see directly from this figure that the repair kinetics of germ-free mice are strongly impaired when compared to SPF mice. So I hope that I've been able to convince you that it seems that microbiome not only regulates epithelial barrier permeability genes, but it directly affects a function of epithelial barrier permeability in a murine host. But the question is, how do commensal microbes regulate epithelial barrier permeability? When we had compared our epithelial transcriptomic data sets, we found that microbiota seems to regulate expression of a xenobiotic sensor called aryl hydrocarbon receptor. We found that consistently the aryl hydrocarbon receptor genes were upregulated in SPF mice. And when we follow this pathway, which is a transcriptional regulator, we found that several genes which are associated with this pathway were upregulated in SPF mice. But importantly, what was key to the observation was a typical prototypical transcriptional response to AHR, which is a xenobiotic sensor, is cytochrome P450 enzymes, which we saw upregulated in SPF mice. This pathway has got a lot of attention in the skin field in the last decade or so, because not only is this promiscuous pathway activated by several environmental modulators in the skin, it is directly implicated in epithelial differentiation as well. So then I sought to see that is there any significance of perturbing this pathway? So we took a keratin 14 Cree mouse in which the Cree promoter is under the control of a keratin 14 promoter, and then basically knocked out this gene specifically in the skin of these mice. And what I found was that loss of AHR, specifically in the skin, did affect barrier recovery kinetics. So shown here in red are mice which do not have a functional AHR in the skin. And you can see that over time, their barrier never really repairs. Then thought, what if we activated AHR signaling in germ-free mice? And to do that, I basically activated AHR signaling using a well-defined ligand of the aryl hydrocarbon receptor pathway, which is called FIXZ. FIXZ is an interesting tryptophan metabolite. It is typically produced when UV acts in skin. It is an indole family tryptophan metabolite. And basically, we use this to activate signaling in germ-free mice. And we did see that FIXZ could improve expression of AHR in these mice. But what was interesting was that activation of AHR signaling actually improved barrier recovery in germ-free mice. This basically suggests that indeed AHR could be a way by which we could repair skin barrier defects in germ-free mice. But the question is, how do commensal microbes regulate AHR signaling, which seem different in these mice? Um, Now, there is precedence from the gut microbiome field that microbes produce tryptophan metabolites, which then act as regulators of AHR signaling. It has been shown through a wide variety of studies that, at least in the gut, microbiome derived tryptophan metabolites regulate barrier function. There's also precedence to look at tryptophan metabolism in skin diseases. So we know that in atopic dermatitis, in some studies, tryptophan metabolism is attenuated in atopic dermatitis. And another disease which is associated sometimes with barrier dysfunction is a disease called hidradenitis suppurativa, where it was found that microbiome of patients with this condition showed dysregulated tryptophan metabolism. So I sought to test the hypothesis that commensal microbes regulate skin barrier through tryptophan metabolites by regulating this aryl hydrocarbon receptor pathway. And this essentially involved developing a novel ecological framework. So far, what I have shown you was work looking at germ-free mice versus specific pathogen-free mice. But in order to really understand how some of these mechanisms would work, we thought that we would use an experimental framework in which we would use a notobiotic model where germ-free mice are colonized with defined skin commensal communities, use that skin and perform targeted mass spectrometry to identify microbial metabolites of tryptophan, and then validate them in vivo as a framework to test if these metabolites can be used as therapeutics. So, in step one, we basically developed a brand new novel notobiotic model to study skin microbiome. And to develop this model, we took this extremely detailed approach in which we wanted to expand our culturing of commensal isolates from the skin. This essentially involves surveying a large number of healthy human subjects and then directly streaking human skin swabs onto 15 different kinds of media and then basically recovering novel looking isolates. And what we were able to do so with this approach was that we were able to curate a 50 species commensal microbe collection, which represents the human skin microbiome, which is depicted here. Essentially, what does this microbiome collection consist of? We call this the Flowers Flora 50 because this work was done in collaboration with a colleague named Louise Flowers. And what this 50 species collection consists of is members of a wide variety of genera of skin microbes. But what you can notice here was that a lot of microbes that we recovered were from Corynebacteria, Micrococciae, and Staphylococcus. What we basically did was colonize germ-free mice for 1 month with these commensal communities, and then basically compared barrier recovery kinetics. That the skin microbiome community colonization indeed improved skin microbiome barrier in germ-free mice that were colonized with this FF50 community. We then collected skin from these mice and performed targeted mass spectrometry to identify the proof of concept. What we also did was take a genomics approach in which we mined the genomes of all these metabolites for tryptophan metabolizing enzymes. And I'm going to show you one small snapshot that when we basically looked at, say, 5 members of this consortia, we found a lot of motifs in the tryptophan metabolizing pathways. However, what was interesting was we saw that a lot of metabolites belong to this cascade, which involved indole acetic acid processing and indole propionic acid. These pathways have been implicated in soil microbiome studies, but are lesser studied in the skin microbiome context. In fact, they have been virtually unexplored. So when we performed targeted mass spectrometry, what we found was that we could detect a wide variety of tryptophan metabolites in the murine skin. So if we looked at germ-free versus Flowers 50 colonized mice, we actually saw a wide variety of metabolites. But what was interesting was that those of the indole acetic acid axes, which included indole pyruvate, indole acetic acid, and indole 3 aldehyde, were significantly upregulated in the flowers 450 colonized mice. I then just wanted to test if these metabolites could indeed improve barrier in vivo. And for that, I use this model, which is an atopic dermatitis model, in which basically this model recapitulates a lot of conditions of atopic dermatitis, in which we perform a tape strip and then do ovalbumin sensitization to recapitulate atopic dermatitis like responses. So in this case, we basically pre-treated the metabolites and then also treated them with ovalbumin and then measured barrier recovery. So, as a control, this is how such an experiment would look, which is that a mock control would basically show where we do not have any ovalbumin, we don't see particularly a change in tool, but when there is ovalbumin control, we see a worse barrier condition. So now with this range, we could measure if the tryptophan metabolites actually attenuate the ovalbumin-induced barrier damage. So what we found is that a variety of tryptophan metabolites improved barrier repair in vivo, but we again saw that the ones which were in this indole acetic acid axis did it to the best level. So what I then did was basically make a cocktail in which we basically had these compounds and actually saw that in that model too, this cocktail could help improve barrier resilience in an AHR dependent manner because when we had an AHR specific inhibitor, this property was lost. I then went back to the colonization model and treated the flowers 50 colonized mice with and without the inhibitor. And we saw that when we had the inhibitor condition, actually that affected the barrier resilience, showing that these effects are AHR dependent. So, what I have for you is basically an ecological framework. So, we started out with a fundamental observation using germ-free and SPF mice that we found these genetically regulated pathways. And then we basically used a culturomics approach to curate synthetic microbiome communities, used a defined consortia to colonize notobiotic models, and then have used a variety of multiomics approaches to identify metabolites of interest, and then do a functional validation of these metabolites to basically see if we can develop therapies for barrier dysfunction. The next steps, of course, involve that can we then harness all this information for microbiome derived precision therapeutics, which can be used to manage skin barrier diseases. So, one approach would be to evaluate therapeutic efficacy of these metabolites. The other aspect, which is critical, is also to dissect mechanisms. And finally, can we actually design probiotic communities which can prevent barrier abnormalities? So hopefully that leaves you with a lot of food for thought for how skin microbiome can be studied and harnessed to treat diseases. In the end, I'd like to present my acknowledgments. So, a lot of the work that I showed today was actually done in my postdoctoral lab, where I was until January of this year. That would be Dr. Elizabeth Grice's lab at the University of Pennsylvania, where I conducted a lot of this work. Subsequently, I have started my own independent group at the Washington University School of Medicine. This is my brand new lab with highly talented and enthusiastic individuals that I am having a huge privilege to mentor and work with. And what we're hoping is that we're going to study more functions of how commensal microbes can be used to treat barrier dysfunction. I invite you all to explore our lab website to learn more about our projects. And I thank you for the opportunity to present my work. And thank you for listening to us. Thank you so much.